So today we're sort of stepping back to DNA, and we're going to examine DNA, and we're looking at it because it helps us to understand DNA as we start to move in cellular replication, mitosis, and then meiosis, the formation of our germ cells, the sperm and the egg. So with DNA, of course, we're back to our slide that we're really starting to move toward now. Where do you find DNA in the cell? In the nucleus. Where else? In the mitochondria. If we're a plant, you also find them in the chloroplast. But principally for our focus, we're going to be looking at DNA in the nucleus. Now, when we make a copy of DNA into RNA, what trans is that? Transcription. RNA is going to be used to encode for amino acids and assemble them into proteins, and translation. Say this over and over again. By the time we get there, it might stick. But those are the two big key events that we're really moving toward at this point. Now, we already know that there are 4 bases that comprise DNA. We're going to leave uracil out. That's for RNA. But we have our A, T, C's, and G's. And we place them into 2 groups, the purines and the pyrimidines. One of these has a single ring structure, one has a double ring structure. How do we remember which is the biggest? Purines are the smallest name, and they have the biggest structure. Pyrimidines, the biggest name, smallest structure. That's how we can understand them. Why is that significant? How are you going to pair your nucleotides? You're going to have a pyrimidine and a purine together in the pair. That's going to span the space between the 2 strands of DNA. So A and T will always be together, and G and C will always be together. Now, how do we remember which of our nucleotides are purines versus pyrimidines? Cut. Cuts are the pyrimidines. So C-U-T, uracil being an RNA, are the pyrimidines, and the others are our purines. Now, there was this guy named Chargaff, and early on, as people were looking at biochemical molecules, looking at trying to decipher what is this inheritable information and where does it come from, they began to look at DNA, and Chargaff said, you know, what's very interesting is that I find very similar percentages of these nucleotides in cells. And so when you look in a cow, there are 28% A's, 28% T's, 22% G's, and 22% C's-ish. So Chargaff said, you know what? Species is really going to be fairly similar. And the A's and T's are going to be similar, and the G's and C's are going to be similar. We didn't understand the structure of DNA at the time. So Chargaff was the first one that says, you know, DNA varies from species to species, but within a particular species, we're always going to see our A's and T's and C's and G's being equivalent. And we know the reason for that, because they're always paired. So that came up to be Chargaff's rule, the fact that A's and T's and C's and G's are going to be the same percentage in each one of our cell types, and that the percentage of A's and T's and C's and G's will vary between species, which makes sense because we're similar to, but also very different from cows. Now, when we look down at plants or we look at some invertebrates, we can see that that species difference varies even more greatly. So the percentage of these is species specific, but we can still look at our A's and T's and our C's and our G's. And if you know the percentage of one, you can calculate the percentage of all of them, right? You understand how we do that? Let's say, for instance, I were to tell you that in a particular species, we had 24% of our nucleotides were A. Determine the percentages of the other nucleotides. What would you do first? You know that T's equal 24. Now, how do we determine the other two? What leftovers? Okay, so what do we got here? 48, what's left over? Divided by 2. So 26% for the C, 26% for the G. Everybody see how we do that? Fairly simple math, but you have to know the relationship. Because those four are going to represent the 100% of our DNA. Now, as we move into DNA replication, that is going to be the very first thing that happens as we start cell division. We're going to see that our two strands of DNA are each going to represent a conservation of code for the offspring. One daughter cell gets one strand, another daughter cell gets the other strand. So each daughter's getting a set of the original instructions. But daughter cells are still going to be double-stranded, so you make a copy from that so that you too as a cell can have double-stranded DNA. Well, we're going to look at Watson and Crick, 1953. That's where they published in Nature the double helical model of DNA. We know we're going to add another person in there. You remember her name? Rosalind Franklin. Yeah, we're going to go into a little more detail about her. In fact, there's her name there at the bottom. So we've got our backbone, our sugar phosphate backbone. We've got our nucleotides spanning between, and what holds the strands together? Hydrogen bonds. Which pair has the strongest bond? Because the bond across is one purine, one pyrimidine. So which pairing? The CG, because there's 3. What does the AT have? 2. Only 2. So that's going to be stronger. So we already know that structure from what we've done before. We don't have to continue to beat that horse. But when we look at Rosalind Franklin's work, this is where we want to spend a little bit more time. So Watson and Crick won the Nobel Prize for their discovery of DNA in 1962, 4 years after Rosalind Franklin's death. She died at age 37 from ovarian cancer. And when you look through her story, it's so very interesting because she received her doctorate in physical chemistry, which means she was really smart, right? If you've ever heard of physical chemistry. She got her PhD in 1945. Why is that significant, 1945? That's right in the middle of World War II. So she got her doctorate when the world is basically falling apart. But then she ended up in England in 1951, and she was a research associate of John Randall's King's College, Cambridge. Pretty good school, right? Yeah, one of the tops in the world. And in Randall's lab as a research associate, she ran into this guy, Maurice Wilkins. And they had separate research groups, but they were both working with DNA. So Randall, who is head of the lab, gave Franklin responsibility for her DNA project. Well, Wilkins didn't realize she was her own researcher, just thought she was a lab tech. And so Wilkins took it upon himself to basically show Watson and Crick one of her X-ray crystallography pictures, that X-ray picture that we see that looks like an X. So he just took it off her desk without asking her. Well, once they saw it, they knew exactly what they were looking at. Unfortunately, even though Franklin's work on DNA in the structure and that X-ray diffraction map, it appeared in the exact same issue of Nature at the same time that Watson and Crick published their paper, she didn't receive any credit for it. She only received credit for it much after her death. I think it was 10 years after Watson and Crick won the Nobel did they finally come out and say, you know what, we need to thank Rosalind Franklin as well. That's just Man, I don't even know what to think about people like that. But we know people like that, don't we? And especially when power, prestige, and money is involved, that's where you're going to get a lot more like that. So once again, we look at our DNA helix. The reason that we have a purine and pyrimidine in the middle is because of the spacing. Two purines would be too big to fit. Two pyrimidines would be too small to fit, so that's why you have one of each. And again, as we said, held together by our hydrogen bonds. Now, as we look at this structure, again, a lot of people compare it to a spiral staircase. You're going to see that there is a larger space between strands and in a tighter space. These are called grooves, the major and minor groove. And this is going to be particularly important when we look at certain regulatory molecules being able to bind in this area in a sequence-specific manner. Most of that's going to be happening in the major groove where we have more space. Now, when we talked about polymerization reactions, monomers being assembled into polymers, one very important aspect of this process was a directionality. Amino acids have an amine group and a carboxyl group. That's going to orient them in the right way. With our nucleotides, we're going to assemble them in a direction that is referred to as 5' to 3'. We call that the reading frame. But since we have two strands, notice what's happening. Our 5' to 3' of our strand on the left is moving downward, but where's our 5' to 3' on the right? It's moving upward. It's moving in the opposite direction. So with our two strands, we say they have an anti-parallel orientation. Now that's really going to be important when we get to DNA replication. One of these strands, we can form a new one in one direction with one mechanism. The other strand, we're going to have to do something different because it's going in opposite directions. We can't synthesize them both in the same direction because the template that's going to be used is running anti-parallel in opposite directions. Now, our typical DNA that we see in all of our illustrations, it is a right-handed helix. Now, we went through the L and the S and the R and those amino acids, you know. Well, here we call this B DNA. Where we came up with the letter B for this, I don't know. Makes me wonder where A DNA is. You would think the first. Well, we contrast B DNA from a left-handed helix that is called Z. And I think you can imagine where we get Z from because you can see how zigzag some of our phosphate backbones look. It's going to give a much more open space, and it's thought possibly that zDNA may occur when we are trying to transcribe transcribe the DNA into RNA, but we're still not really sure. This is the more common structure. This one can occur. So bDNA is naturally occurring DNA, and we'll just leave it at that. And bDNA stands for the right-handed helix. So here we have major, minor groove. Major groove, again, this is where we are usually going to read the sequence to copy into RNA. The minor grooves may give access for proteins that will interact with the DNA, either regulatory proteins that we call transcription factors. We'll talk much more about those when we get to DNA replication. But we also have proteins that are going to bind to the DNA to help it be packaged. And so that's going to be important in those spaces of the minor groove. It's just more difficult to get to the nucleotides to read those. Now, when we look at our DNA molecule, we're going to see that each chromosome we have, if you were to stretch it out, it will probably reach about 5 centimeters. Now that's on average. Some might be longer, some might be shorter. So we have these linear right-handed helical molecules that we call chromosomes. Bacteria, of course, have circular DNA. And in our circular DNA, this is a beginning place where we can look at how coiled our DNA becomes as it gets packaged. Now, they used to have these a lot, the old telephone cords that were the spiral telephone cords. Anybody ever played with one of those? My granddaughters were playing with an old handset that had one of those you plug into a smartphone, and you can actually use it to talk with. My girls love talking on that. But if you've ever spent any time with this thing, especially back in the day, you know that that coil, if it gets a certain amount of tension on it, it will start to twist around itself. Have you ever had that happen with electrical cord? And so what you would do, and it would drive me crazy, I would hold the handset down, let it unspin to release the tension. So when it was straight, there wasn't a lot of tension. But when you twisted and twisted and twisted and it started to twist back on itself, that's what we refer to as supercoiling. We have a circular DNA here, and you can see if you apply enough twist, it's going to twist around on itself like that phone cord. And it can either be in one direction, which is negative, or in the opposite direction, which they consider positive. But this is the supercoiling we can happen. Here we can see a plasmid that's relaxed, and here we can see a supercoiled plasmid. Now, our linear DNA is also going to have twist. It's going to have supercoiling, even though it's a linear and not a circular structure. But we're also going to use proteins to help this happen and to package our protein into more and more condensed structures until we can see them under a light microscope, in which case a lot of times those X-shaped things, people call them chromosomes, they're actually sister chromatids. That's just our DNA, just super compressed down. Now, since our DNA can exist in a twisted state, in our phone cord, we can just let the handset or whatever, or disconnect it and let the cord unwind. DNA, we can't do that. So we have enzymes that are called topoisomerases. Big fancy name. I like to call topoisomerases untwistases, because that's what they do. They untwist the DNA, remove the tension. And so instead of just uncoiling it, what they do is they will make a cut in the DNA, allow the DNA to twist to remove that tension and put it back together so that it's not coiled again. And there are two topoisomerases, 1 and 2. Guess what? The topo 1 and the topo 2, how they're different. Topo 1 makes a single-stranded cut. Guess how many cuts topo 2 makes? 2. It cuts both the strands and passes the whole thing over to remove the tension. And that's the only difference. That's how they untwist. Topo 1 with a single strand cut, topo 2 with a double-stranded cut, and moving the DNA to remove the extra twist. Now, we're going to see a lot of enzymes as we get into DNA structure. Topoisomerases is just an important one. You have to remove that supertension, that supercoiling, before you can begin to do the rest, like the unzipping with the enzyme we call helicase. Now, we've already touched on this a bit, because when we talked about the bonds that hold our double strands together, what did we say they were? Hydrogen bonds. Which pairing has the strongest bond? CG, because they have how many? 3 hydrogen bonds. AT has 2. So if you look at a region of DNA or a chromosome, if you have a lot of CG pairing, it's going to take more energy to separate those 2 strands. If there's a higher concentration of AT, it'll separate at a lower temperature. Now, we can do it with heat energy. You can boil DNA and you can denature DNA. Denaturing means the two strands separate. And we can do that because our hydrogen bonds, are those the strongest of our bonds? No, which is the strongest? Covalent bonds are the strongest, hydrogens are the weakest. So we can use heat energy to denature them. And as you look at our temperature of our DNA, typically when we're running experiments, especially when we get to our PCR experiments that we're going to talk about a little bit later, when we need to denature the DNA, we do it at about 94, 95 degrees centigrade. Boiling is 100. So we're close to boiling. It's a lot of heat energy. And when you look at a typical standard DNA molecule, that melting temperature when we're going to start to unanneal, move the DNA strands apart, that temperature is going to be largely based on the ATCG content. But the melting temperature of DNA is where the two strands, you break the hydrogen bonds, and the two strands separate into single-stranded DNA molecules. Now what's really cool is when you heat them up like this, if you lower the temperature, they'll just slide back together because of the base pairing. If you've got a lot of nucleotides that are the same and start to form, then it'll just zip right up. If you start to get a mistake and the next one doesn't match and the next one doesn't match, then it'll just come apart again. So DNA can more or less spontaneously come back together. So once again, here we see more or less an experiment where we heat our DNA up. You see we're going up to 90, 100 degrees. So we've completely melted our DNA. We've gone from double-stranded to single-stranded DNA. Then at this point, we start dropping our temperature. You say cool it down to 20 degrees, room temperature-ish. And over the course of 30 minutes, even less than an hour, we have completely renatured, see this is denaturing, renatured or reannealed our DNA strands back into that double helical structure. How much time? Depends on how long your DNA strand is. Longer strands take longer time. Make sense? This is important because this characteristic of DNA is going to be critical for our manipulation of it in a lab. We're going to get into two techniques that we use in a lab that makes that extremely important. So we talked about the ATCG. Here is a practical example. As we increase our GC content, we see bacteriophage, salmon sperm DNA, calf thymus, there's E. coli. As we look at almost a straight line of our different species, we find that the higher the GC content, the more temperature, the more energy is required to break. And this increase in temperature is directly related to that GC having the three bonds and needing more energy to separate. The higher the GC content, the higher the temperature needed to denature the DNA. Because as you add GCs, you're adding one additional hydrogen bond that the AT pair didn't have. So it takes more energy to break. See, I'll review so far. Have we hit anything really new yet? Major groove, major groove, minor groove. But that's about it as far as new material. So far, we're still reviewing. So now let's talk about genomes. What is a genome? If somewhere off the street were to ask you, what's a genome? What would you tell them?
DNA makeup of a species.
All your DNA. All your DNA, that's a genome. Now, we like to think we're special as humans, right? The most advanced species on the planet. Wouldn't you think we're the most advanced species? We would have the largest genome of any organism on the planet. Don't we need more DNA to be so smart? No, it's actually not the case. Yes, oh no, I love that. I love that. And in fact, when we look at how much DNA we have, we got a lot more than we ever use, which makes me go, why do we have it all? And a lot of people are theorizing about that as well. But where are humans in here? Here's mammals. You think mammals, pretty good size genome, right? Protozoans. There's some protozoans that have ridiculously more DNA, more nucleotides in their genome. Plants have more than we do. Amphibians have more than we do. Now, viruses have a pretty good bit. When you think about the size of a virus, that's still pretty impressive. Viruses aren't even one single cell. We have trillions of cells, and they've got a pretty robust genome. Now, we've already talked about the prokaryotes, circular DNA, plasmids, eukaryotes, linear DNA, and then we've also got that circular DNA in our mitochondria and plastids, the least of which is the chloroplast that we talk about in plants. So, an amoeba. Is amoeba very advanced? I wouldn't think we would think an amoeba is very... 200 times more DNA than humans have. That just kind of blows my mind. So I guess more isn't always better? I don't know. But I can't wrap my brain around the significance of why some of these organisms have so much in the way of DNA, even so much more than humans. Yeah, Miranda, I like your work. Harder, not smarter. That's the human motto. Smarter, not harder. Oh, did I get it backwards? Yeah, smarter, not harder. It's Monday. All right, now to get into some molecular biology. Bacteria are really good at moving DNA around. And they can integrate their DNA with other DNA. So bacteria produce enzymes, proteins, that cut DNA in very specific sequences. And we call these sequences restriction enzymes, and they cut at their restriction sites. Now, typically, we're going to see that the target of these restriction enzymes is going to be in the neighborhood of 6 nucleotides long. Some may be as short as 4. And now for the molecular biologist in a lab, if you're needing to work with DNA, you can look in a catalog and you can order from a series of hundreds of restriction enzymes that have been isolated and are commercially available for your use. And they usually have some interesting nomenclature. Here we have an enzyme HAE3, HAE3. And we see that the restriction site is a GGCC. You see our 2 strands, the parallel, the anti-parallel strand. And when the enzyme recognizes those 4 nucleotides, it cuts right down the middle. And it breaks that strand. Bless you. We call this a blunt-ended cut. Here we have one small one. It recognizes CCC-GGG, that's 6. It also cuts right down the middle. So these 2 enzymes recognize completely different targets.
We need to be able to recognize those names.
No. What I want you to know is the blunt-ended cut versus what is called a sticky-ended or a cohesive-ended cut. You see the difference, right? So when we look at, one of the enzymes I used a ton, to me it's one of the more commonly used ones. It was in the lab I came from. Echo R1 recognizes GAA-TTC. When it recognizes this, and notice the sequence is something called a concatemer. Do you know what a concatemer is? What's a doppelganger? You ever heard that one? Your evil twin. It's like your opposite twin. Well, think of a concatemer as a sequence that has an opposite evil twin. Because look, here's GAA-TTC. Look down at the bottom. GAA-TTC. It's the same sequence but backwards. That's a concatemer. And when the enzyme recognizes that sequence, it doesn't cut right down the middle. Look at the staggered cut that it makes. So for molecular biologists, when I was working with plasmids and working with genes that I wanted to insert into a plasmid, I much preferred a sticky-ended cut because these have a much better chance of only annealing with a corresponding cut. If you get a blunt-ended cut, some of these can just go back together because they're flat. This is going to be much more specific for what you can put together. Now, these are also referred to as endonucleases. Endo means it cuts within the strand. An exonuclease has to find the end of a strand before it can cut pieces off. Do you see the difference? Exo from the outside, endo inside. And so we use these restriction enzymes to tell us a lot about the DNA that we're investigating. So when we make our cuts, we usually take the fragments of DNA that result from those cuts and run them out on gel electrophoresis, and you separate out the strands based on size. The smaller strands go toward the bottom. The larger strands, it takes longer for them to move through, so they stay at the top. And depending on your banding pattern, when compared to a sample of known lengths, you can get an idea for where your restriction site is within your DNA strand. So we'll give you an example. So here we have a piece of DNA. We know our piece of DNA is 7 kb kilobases long. So here's our uncut DNA. It stays at the top, at about the same place where our standard size would be 7. That's right. And if we cut the sample with Echo R1, we see that we get a 4.5 fragment and a 2.5 fragment. So if you start with one piece and you end up with 2 pieces, how many cuts did Echo R1 make? It made 1 cut, somewhere in that strand, cutting it into 2 pieces. Now we come look at Hey3. We use that enzyme and we see 1, 2. We get 2 fragments. How many cuts? 1 cut. So when you look at Hey3, we got a 5.5 cut, and we got a 1.5 fragment. Echo R1, we had a 4.5 and a 2.5. So when you look at these fragments and then you cut with both and you end up with 3 pieces, 3, 2.5, and 1.5, which map below would most represent where the restriction sites would be? See, here would be a 2.5 cut. There would be, see, that's 2.5. There's 5.5. There's a full 7. So we've got our 1.5 here. We've got our 3 here. And we got our 2.5 there. That would fit, right?
So are we just reading it from the top one? Is that what we're doing?
Yeah, so, they didn't show you, but when you run these bands out, you've got another lane that's got a bunch of bands in it, and you know the sizes of those bands. It's called a ladder. Okay, and they just know that at this point, that would be 5.5, this point, 5.5. So, because that band ran further down, it would line up with our standard, that would be 3 kb. That would line up with our standard such that we know that's 2.5, and that's 1.5. So we know we got 2 cuts within the strand, so where's our cut going to be? Well, if Hey3 cuts here, that's going to be a 1.5, right? Would this be the best place for Echo R1? Is that where we would think it would cut? Because see, Echo R1 gave us a 4.5 fragment, so maybe it cuts here. So which of these is correct? A? Why? Because when you cut with both, see, we're cutting with both here. Echo must be at that site given a 2.5 and a 4.5, because that's what we got here with our 2.5 and 4.5. it couldn't be here, because if it was here, we would expect when we cut with both of these that somehow we would end up with a 1 KB fragment and still have a 4.5, and we don't. You see, that's a little bit of detective work. And so from all 3 of these lanes, we know that this would be the pattern of our restriction sites. Echo cuts here with a 2.5 and a 4.5, and Hindi 3 is going to make a cut in this 4.5 fragment that Echo R1 would leave over, dividing it into a 1.5, a 3, and a 2.5. Not getting it?
No.
All right.
I remember this from genetics.
So Echo R1, we get a 4.5 and a 2.5 cut, right? It cuts one place. So ignore the Hay 3 in this picture. That echo could cut on this end to give you a 2.5 and 4.5, or it could cut on this end and give you a 2.5 and a 4.5, right? Hay 3, we could say that it could do the same thing on either side. But the author in this particular picture is telling you that they know that's where the Hay 3 cuts. Which of these would be correct? Here, you would have a 2.5 fragment. From here to here is a 3, and from here to here is a 1.5. Down here, if this was the pattern, you would still expect a big 4.5 KB cut, but you don't. It's not there. So this one has to be the right pattern. Okay? Again, it's just detective work and sort of accounting. Where are the pieces? Pieces and parts. Now, the restriction mapping also, because of those cut sites, allows you to glue separate DNA pieces together when you're manipulating them. But DNA fingerprinting, a lot, is built on restriction enzymes, because different people are going to have different sites in different places and different sequences between, which are going to give them different links. It's a pretty easy way to do that. Now, this is a big one. PCR, polymerase chain reaction. We already know what polymerization is, right? Taking monomers, making polymers. And so polymerase chain reaction is going to involve making new strands of DNA from a template. The chain reaction part means we're going to do this over and over and over and over again, maybe 35 times inside of a test tube, so that we are going to exponentially amplify a specific region of DNA. So this is how it works. We're first going to take our template strand of DNA, double helix, right? How can we separate the two strands? Heat it. You heat it. And here we're heating it up to 94 degrees, and we denature the DNA. Now, inside the solution that we're doing this in, we have also added little synthetic pieces of DNA that we've ordered from a company. We call them primers. And these primers are specific for two sequences in our DNA. One of our primer is going to bind to the anti-parallel strand. Do you see how that little short piece bound here on the right side? And at the top, we have another piece that's going to bind to the parallel strand, and it binds here. So you have two primers, one on each end. So we anneal the primers, and all these primers swimming around in our solution prevents the big strands from getting together, because remember, shorter strands anneal quicker than longer strands. So that's step 2, annealing of the primers. Step 3, we allow an enzyme that's also in the solution to begin to assemble our nucleotides that are also in the solution, assemble them together into a brand new DNA strand. So in our PCR cocktail, we have our template DNA, we have our forward and reverse primers, we have our nucleotides. We call them DNTPs because we have A's, T's, C's, and G's. But then we have an enzyme that is called Taq polymerase, T-A-Q, write that down, Taq polymerase. Actually, I think it's written T with a subscript AQ, Taq DNA polymerase. The Taq stands for Thermus aquaticus. It's isolated from a bacteria that live in these hydrothermal vents, these hot springs. And so what is the cool thing about this enzyme? When you heat it to 94 degrees, it won't denature. Its structure is such that it's going to have to be a lot hotter to denature. So you can then, after you go through one cycle, you can heat it back up, the original template, and now your two new templates denature, cool it down, come back together with the other primers, and it goes through this all over again. That enzyme will keep doing this over and over and over. So do you see the exponential expansion? You start with 2 strands, your template, you go through a cycle, now you have 4. You go through the second cycle. Each one of those is going to be double. You go from 4 to 8, 8 to 16. You see how we're going up, we're doubling each time. That's the beauty of PCR, and what makes it possible is that Thermus aquaticus DNA polymerase, because it doesn't denature when you heat it back to 94. So here we denature. Watch our strand come apart. There's our little primers, and there's our Taq polymerase synthesizing our new strand. And again, you do it over and over. Denature at 94. Cool it down to 54, 50-ish, and that's going to be specific for your primer template strand. And you make a new strand with Thermus aquaticus and do it again and again and again and again. First cycle, second cycle. By the time you do 35 cycles, you have made 68 billion copies of your DNA. And the copies that you're making, it's just going to be the DNA that spans between where your forward primer and your reverse primer are. That's what you're going to expand 68 million times. So this way you can expand a very specific piece of DNA. It's usually not going to be an entire chromosome. It's going to be a small fragment of DNA that you can amplify, use in various experiments, get sequences from. In the lab in the Cancer Center in Arkansas where I work, we would use primers to produce genes, but the primers would have built-in restriction sites or built-in mutations, so that as you made a new gene, you could cut the ends and you could put it right into a plasmid with those sticky-ended cuts. So you engineered a restriction site in because they were built into your primers. Or you could build your primers in such a way that in these places, when you made the gene, you changed a nucleotide. And if you change one nucleotide, you can encode for a completely different amino acid. So there are a lot of things you can do with PCR, not just amplify for sequencing. I mentioned sequencing, which is another thing that we can do. And in a sequencing reaction, it's going to be very similar, as you see, to what we're doing with PCR. The only difference is we don't have to use Taq polymerase because we're not doing 35 cycles. And in addition to having our DNA polymerase, our nucleotides, we also have a different set of nucleotides. You can't see these, but over here they're represented by the triangles. Normally our nucleotides are D-NTPs, deoxy ATP, deoxy TTP. But in this case, we have dideoxys as the triangles. The dideoxys can be inserted into the strand, but they can't have anything else added to it. So whenever you stick a triangle end, the polymerase stops, so you end up with a short piece. We're going to have another triangle inserted. Where is it going to be inserted? Where's our triangle? Follow this. There it is. And see, that one's longer than that blue one. And as you do this, just random chance, we're going to have DNA fragments that differ by one nucleotide. And it just so happens that these triangles, in addition to the dideoxy, have a fluorochrome attached to them. So when you hit them with fluorescent light, what are they going to do? They're going to light up. They're going to light up either blue or red or green or yellow. And when you run all of these strands in your sample through basically a computerized chromatography machine, that computer is going to be able to tell you your sequence of your DNA because they differ by one nucleotide. So in the past, when we just used radioactive markers and did this experiment, we would have to look at these gels and look at the little bands, and we would have to do it ourselves because you had to run 4 lanes, and you would look, okay, well, here's a band in the T, and here's a band in the A, then here's a band in the G. Now you just send somebody your sequence, your DNA, in a day or two, they send you back a computer email that's got your sequence all mapped out. Very quick, very easy. It costs you, but hey, work smarter, not harder. Like that. But do you see how the sequencing works? It's all because of the dideoxy colored nucleotide and the fact that they stop at one sequence, and then a computer can tell you this one, then the next longer one is this one, then the next longer one is that one, all based on the color of those nucleotides. So, I mean, this is already outdated. There is no telling how many genomes have been sequenced because they have robots that do this now. Robots and computers. I don't know humans do much of anything other than load the sample in the machine. And these machines run night and day to give us our sequence. So our nuclear sequence for humans, we're around 3.2 billion nucleotides in the human genome. We're 1,000 times what E. coli has. We're not talking about amoebas, right? Amoebas got us beat. It's hard to believe the Human Genome Project was completed in 2003. It took decades to get the human genome. Now you can send a species in, and you can get a genome from a species in a few days. If that. It may be down to hours now with the increase in technology.